somewhere? Weird. Okay, good morning everyone. Nice to see you again, and thanks for those who've come today. We're continuing on this pretty important topic of pharmacogenomics in this lecture, and remember that we first, in my lecture on Tuesday, addressed the why this topic matters, because inherited variability in genes for proteins and enzymes that control the fate of small molecule drugs in the body have a big role to play in shaping your clinical response to pharmaceuticals, and it can have an influence on how effectively the drug reaches the target organ for pharmacological effects. So let's say it's a drug used for treating depression. The ADMET processes control concentrations of drugs not only in the most accessible biofluid in blood, but also the spectrum or the profile of concentrations in the target organ. But then, of course, they also influence the level of exposure of sort of off-target tissues from a pharmacological point of view, organs that don't really have any role to play in terms of mediating the clinical benefit for a person, say suffering from depression. But nonetheless, they can be important targets for toxicity caused by drugs. And so a good example would be something like skeletal muscle. Certain drugs have the ability to cause myopathy and muscle injury. And that has something to do with the concentrations and the profile of muscle concentrations over time. But of course, a really big role where drug metabolizing enzymes and transporters play a key role in pharmacology is in enabling those critical excretory organs, the kidney and the liver, to actually remove drugs from the body and metabolize them or permanently get rid of them. Okay, so any inherited deficiency in some of these pathways, critical, if that particular protein or particular enzyme is important for a particular drug, then there will be clinical manifestations of a pharmacogenomic defect or deficit. Now, we're going to particularly focus in this lecture on a very important pharmacogene and one that's probably the best research from a clinical pharmacology point of view, perhaps. I don't know if that's strictly still true, but that's cytochrome P450-2D6, which is probably of all the P450s that contribute to human drug metabolism. You can see in this pie chart there's a whole bunch of them. Many of them are subject to polymorphism, but in terms of 2D6, polymorphism really does matter. And you can see the author of this slide, they've put polymorphism in bold here for that, next to that yellow pie slice for 2D6. And they've also done that for 2C9 and 2B6 and 2A6 here. But for a lot of the other P450s, the polymorphism is highlighted in unbolded text, just showing you that it exists, but it's the ones that are highlighted in bold that are particularly clinically relevant. And that's why I'm focusing on 2D6 in this lecture, because it's so important in metabolising a lot of pharmaceuticals. And the other thing that's interesting about 2D6 is, as you can see from all these other P450s, a lot of them are subject to induction or downregulation in inflammatory states. And I think I talked about that in my lectures on P450 last semester. But you can see for 2D6, a bit of downregulation and inflammation, but there's really no issue with P450 induction with 2D6. And this is one of the interesting things about this particular P450. Its activity is determined at the genetic level, at the pharmacogenetic level, and whether you're exposed to a P450 inducer doesn't affect its activity much at all. So we can remove that sort of pathway to clinical significance. Okay, so why is 2D6 then? So what I want to do in this lecture is talk about an important pharmacogene involved in oxidative drug metabolism. In the next lecture we'll talk about a transporter that's subject to significant polymorphic variation. And then hopefully I'll also have a following up lecture on HLA alleles, lymphocyte alleles, as a source of drug variability to drug-induced toxicity. But today we're looking at P450 as a source of clinical variability to drugs. And the reason why it's so important in pharmacogenomics is really just a reflection of its high importance in clinical pharmacology. This P450 tends to metabolise basic greasy sort of drugs, so anything with a nitrogen atom in a ring usually is often a substrate for 2D6, and that includes some pretty significant pharmaceuticals. You can see on this list here pretty important antidepressants there, very widely used drugs, atypical and typical antipsychotics, even antineoplastics such as tamoxifen gets metabolised to some extent by 2D6, adrenergic blockers, metoprolol to some degree, propranolol, and then down the bottom some very important analgesics where 2D6 has an enormous impact on their clinical effectiveness. Now, why are we considering 2D6 then in a lecture on P450 polymorphism? Well, really, it's partly because I'm getting really old, and when I was a student like you guys, this was really the only one that we knew about was subject to significant polymorphism, and a lot of this early research was coming out in the sort of late 70s and 80s when I was a student, and by the time I was sitting where you were in third year, when I was at 84, I guess there was quite a lot of clinical work being done on 2D6 polymorphism. And back then we didn't know, we didn't even have the 2D6 name, that name hadn't been invented. But we knew about this drug because of two significant episodes involving two cardiovascular drugs. So one is dibrazoquin, another one's spartane. And spartane has significant toxicity issues. It was never approved by the FDA, but the dibrazoquine, I think, is used in Europe. But the interesting thing about dibrazoquine is that it's an antihypertensive agent, and you can see it's getting oxidised by P450 up there in the top right. So it forms this hydroxylation product that lacks clinical efficacy. And you can see the hydroxy group coming attached to the drug there on that ring. So 4-hydroxydibrazoquine. Now, it turns out that some people just can't carry out that particular metabolic transformation. So if you happen to have a particular phenotype, then you'll be unable to oxidise dibrazoquine. Now, the implication of some of this was that in these early clinical trials, when the drug was being studied back in the 1970s, a small proportion of patients enrolled in these clinical trials with dibrazoquine were so susceptible to the drug as a blood pressure lowering agent that they literally had a collapse. They fainted. They had such a profound antihypertensive effect on the patients that they collapsed in the clinic on the floor. It's sort of like, what the heck's going on here? It was just a very small percentage of recipients. So this attracted people to this field, and they began looking for metabolites, and they found that these people that were very vulnerable to antihypertensive effects of dibrazoquine couldn't metabolise the drug. So they looked in their urine at the ratio of the parent drug here relative to the 4-hydroxy metabolite, and you can see these poor metabolisers here. They lack a functional P450 2D6 gene, and so they just can't convert the drug to that metabolite. So these people were called poor metabolisers, and back then it was all done on this phenotypic scale. So they worked out that across the human population there is a range of ratios, and they put people in different phenotypic groups. And we still use these. There are ultra-rapid metabolisers. These are people who extensively metabolise these sorts of drugs by this pathway. Then there are extensive metabolisers where the concentrations of the metabolites are about 1:1 with the parent drug. Then there are intermediate metabolisers who don't metabolise it so well. And then, as I mentioned, the poor metabolisers, the drug virtually comes out almost completely as the parent drug in the urine rather than the metabolite. Okay, so this was very interesting, and people began, and we'll talk about this in a minute, looking at what are the genotypes associated with these particular phenotypes in terms of metabolite ratios in the urine, and it turns out there's lots of them that can contribute to these different phenotypes. So it's what makes this field interesting and complex. And then, as people kept researching this field, some significant things happened. In 1984, the 2D6 protein was purified. A few years later, the gene was mapped to 22q13 on the chromosome. A couple of years later, it was cloned and sequenced. And then there are a couple of pseudogenes found at the same 2D6 locus, and we'll come back to that in a minute. And then a few years later, in 1990, these were the first clinical pharmacogenomics tests. A couple of tests came out to pick up some of the polymorphic variants of 2D6, star 3 and star 4 alleles in particular. Okay, so clinical pharmacology was gaining in expertise in genomics during this period, and still very much a concern in this field. Now, we're going to come back to this in a minute, but like I said, the original discovery involved phenotypic classifications of patients according to the metabolite ratio of the dibrazoquine to 4-hydroxydibrazoquine formed by 2D6, and people were classified as poor metabolisers, intermediates, extensive or ultra metabolisers. And we now know that there's a bunch of different genotypes contributing to each of those phenotypic groups and classifications, and so the people with the ultra metabolisers, these people don't respond much at all to the drugs, or they require massive doses. And this is because they have excessive numbers of 2D6 genes in their genome. They have sort of tandem repeats of the gene. There are a bunch of genotypes contributing to the poor metabolizer phenotype, including gene deletions, but most commonly various single nucleotide variants, and even deletions contribute to that. Now, we won't go through all these yet, but we'll come back to this in a minute. But we'll look at what is it about these variant forms of 2D6 that confer this poor or intermediate status on a person. What is it? What do they do to the P450 2D6 protein to make it screw up its function, basically? We'll look at that in a minute. Now, the other thing that makes this gene interesting, quite distinctive I think, compared to some other P450s, are these so-called pseudogenes, and 2D7 and 2D8, and the P on the end there tells you it's a pseudogene, which means it's not catalytically active or expressed. But because there's a lot of homology with the actual 2D6 gene, this can make analysis of that gene via PCR pretty challenging, from what I understand. I've never tried to do it, but I gather it's very easy to think you're measuring 2D6, but you're actually measuring sequences from one of those pseudogenes if you're not careful with the design of your PCR probe. Now, you're probably thinking, well, why does this matter clinically? What's the point of thinking about pharmacogenomics of 2D6? Well, it certainly does significantly impact the clinical pharmacology of certain drugs, so much so that in the case of codeine, this is the reason why the drug was pulled from an over-the-counter preparation. You used to be able to, probably less than 10 years ago, walk into a pharmacy and get 30 milligram tablets of codeine if you had some strong pain or something. Now, those days are gone. And one of the reasons is, it's for paediatric reasons. There are a number of deaths of toddlers around the world with kids that got doses of codeine inadvertently, and they were in the ultra-metabolizer phenotype, and they churned out tons of morphine. So 2D6 carries out the bioactivation of codeine, so it's a prodrug, and it needs to be oxidised here on that methyl group. I thought there was essentially it's this hydroxylated metabolite, which of course we know as morphine, and that causes strong respiratory depression. So these infants with the multiple copies of 2D6 were very vulnerable to the respiratory depression of codeine. That's what sadly led to the deaths of these little kids. So this is one reason why codeine, in fact the primary reason as far as I can tell, the drug is no longer available over the counter. Okay, so that's an example of high clinical relevance where a prodrug depends on 2D6 to be converted to its active pharmaceutical agent, and so any phenotypic or genotypic variability in that essential enzyme that carries out that conversion is going to be clinically relevant. Another drug where 2D6 shows its significance clinically is some of these old tricyclic antidepressants, not used as much as they used to be, but nortriptyline here, you can see 2D6 is undergoing hydroxylation to 10-hydroxy nortriptyline up here with this oxy group on the 7-membered ring there. And the interesting thing is that plasma concentrations of the parent drug are strongly influenced on your number of functional P450 2D6 genes. So this is everyone's getting the same dose of 25 milligrams of nortriptyline. People without a functional 2D6 gene at all, so both of their alleles are inactive, have very high concentrations of nortriptyline up here, over 50 nanomolar. If you've got one functional gene, then the levels are a little lower. If you've got two copies of functional alleles, then lower again. And then if you've got multiple tandem repeats, some people have up to 13 2D6 genes head to tail in their genome, particularly common in Scandinavia and elsewhere. These folks You can see plasma concentrations from the same dose of drug, very low indeed, and their corresponding impact of on plasma concentrations of the metabolite, the 10-hydroxy metabolite. So if you've got umpteen copies of functional 2D6, you can see very high concentrations of the metabolite here. I think it's still nanomolar, 200 nanomolar, compared to other people with a couple of functional copies of below 50 nanomolar. Okay, so that's just an example of how your genotype influences your pharmacokinetic properties, or the pharmacokinetic properties of this drug. Okay, for the rest of the lecture then, let's just start looking at this issue at the level of molecular structure and function of 2D6. Why is it that there's such variability in humans with regards to levels of P450 activity in a 2D6 isoform? Like I said, this is a well-studied phenomenon. People have been looking at this for close to 40 years, really, perhaps 50 years. And like I said, there's this issue of pseudogenes complicating analysis of 2D6 genes. It took a while for people to figure it out because each of these genes contain 9 exons and have high homology. So you've got to be very careful with the design of your primers if you want to specifically identify the functionally active 2D6. And remember that issue I mentioned in my lecture on Tuesday with the whole idea of a haplotype, where the same gene might have multiple sites in the gene that are susceptible to polymorphic variation, and so people might, when a couple have children, you get this phenomenon of gene shuffling and recombination, and so you can get different combinations of those two alleles in the offspring, and this gives rise, this is particularly an issue with 2D6, and we'll look at some of the manifestations of this in some of the primary groups of 2D6 functional groups in a minute. Okay, so some of the single nucleotide variants occur on a single allele and there's just a single haplotype. So for example, this deletion here at position 2550, deletion of an adenine, it only occurs on CYP3A5 star 3. Well, this is a general comment here, that shouldn't be there on 2D6. But then on the other, you can see some others occur on multiple different allelic variants. So this one here, C to T, 2851, occurs on 2, 8, 11 and 17. 1022, cytosine to thymidine, occurs on multiple haplotypes as well. We'll look at that in a minute in more detail. Okay, so these various variants then have been studied in a lot of detail for 2D6, and if you look at the PHAR website, you can go in there and click on P450 2D6, and I should have done it to update this lecture, but there are literally hundreds of 2D6 variant forms, and all depending on the mutational event involved in generating them. So there's a whole lot that have normal function alleles up the top there, star 1 wild type, but also star 2, 27, so on. And then there's a whole lot of null alleles where there's no activity at all, a whole bunch of those you can see there. And then others confer decreased activity, decreased function alleles. Some confer increased function. Okay, so a whole lot of distinctive genetic events can contribute to different types of phenotypic outcomes in terms of normal function, no function, or decreased function or hyperfunction. And then there's quite a lot of variants that have been identified by geneticists, and we still don't really know the functional impact yet of some of these variants. So this is one of the issues in this field, is that the genetic technology often goes ahead of our pharmacology in terms of knowing the functional consequences of some of these genetic variants. Okay, so I've just picked out a few P450 2D6 variants just to highlight the way that different phenotypes can be conferred by different types of genetic events. So, like I said, this is a year old, this slide, but as of a year ago, just about 150 variant gene families have been identified. So I've just picked out about 7 or 8 of them here. But you can see 2D6 star 1, so that's the normal function form of the protein. And all the various suballeles or subvariants, there are 47 subvariants that are assigned to this star 1 class. They all involve predominantly base substitutions, and they don't alter the catalytic activity, so they have normal activity. And then coming down the list, you can see here star 3, 2 subvariant classes. These are catalytically inactive, so they're null phenotypes, and due to deletions of bases. Splice variants in star 4 groups, a whole bunch of those. Full deletions in star 5, and then single base deletions in star 6, and so on. Okay, so the point is that this gene is really vulnerable to genetic variability more than any other P450, and a whole lot of different types of mutational events can contribute to these different phenotypes. Now, people who worked in this area realised we needed a way to get our heads around all these 150 different types of 2D6 variant forms. And I've come up with this quite simple scoring system. And this is a P450 2D6 activity score. And basically the idea is that we've got 2 alleles of every gene, including 2D6, 2 copies, 1 received from each parent. And we give a score in terms of activity to that 2D6, anywhere from 0 if there's no activity at all the way through to 1 if it's got normal catalytic activity. So that means that if you've got 2 copies of a normal gene, 1 from your mum and 1 from your dad, then you've basically got a score of 2 under this 2D6 score system. Okay, so you can see the example here. Let's say we've got a person with 2 copies of 2 different heterologous sort of gene alleles here. So 2D6 star 1 on one of their P450 genes, normal activity, so they get a score of 1 for that. But then the other allele from their mum, for example, is a star 4 variant form. It's a null form, so it's got no activity, so they get 0 for that. So their overall activity score is 1. On the other hand, this other person, they've got 2D6 star 1 times 2, which tells us that they've got 2 copies of a 2D6 gene, but then the other one is also star 4 null. So in this case, they get 1 times 2 plus 0 equals 2. So essentially, this sort of simple scoring system is the way we work out the activity score for every reported P450 variant gene that gets deposited in a PHAR database. Then hopefully there will be information provided concerning the activity score associated with that particular genetic variant. So in this case, those 4 phenotypic groups that I mentioned were discovered way back in the 1970s by measuring metabolites of 10-hydroxydebrazequin in the urine. People were found that couldn't metabolize the drug very well. Well, those poor metabolizers usually have 2 null alleles, so they just can't carry out P450 dependent metabolism at all, so they get an activity score of 0. Intermediate metabolizers have an intermediate score anywhere from 0 to 1.25 because this system allows for scoring partial activity, so 0 is no function and then 0.25, 0.5 is decreased function, and 1 is for full normal function. Normal metabolizers, people with that phenotype have an activity score between 1.25 and 2.25, and then those people with the multiple copies of functional genes, the ultra metabolizers, have a score in excess of 2.25. So this is just a simple phenotypic scoring system that helps people get their head around the clinical impact of a particular 2D6 gene variant. Now, because we're interested in molecular pharmacology, I guess the question becomes, you know, what's going on in these protein, in the 2D6 protein, to confer this sort of variability in terms of capacity to metabolize substrates? What's going on here? And I just thought for the rest of the lecture I'd focus on this particular study here, which I think was some Australians actually, if I remember rightly. These are some people from I think Victorian College of Pharmacy might have done this research, but they essentially took wild type and 3 common 2D6 variants and expressed them in bacteria, and then looked at the kinetic and structural features of the variant gene, and then tried to work out what impact that had on the ability of the protein, 2D6 protein, metabolize venlafaxine. Remember, venlafaxine is a common antidepressant drug, a serotonin reuptake inhibitor. But also, another area where we see a relevance of pharmacogenomics, it's not just with regards to certain P450s have a poor ability to metabolize a drug. Having a particular P450 variant gene can mean you're not as susceptible, or you're more susceptible to a particular P450 inhibitor. So that variation in the protein structure doesn't just affect ability to metabolize things, it also affects the tendency of a drug to inhibit that P450. So that's another aim in this particular study. They looked at not only venlafaxine catalysis, but also they looked at inhibition by, I think it was quinine, a 2D6 inhibitor. Okay, so what these folks did, they did a wild type 2D6, normal function, and compared it to 3 major 2D6 variants. And they chose these just because they vary according to their prevalence in different ethnic groups, and they're quite significant players in particular populations. So 2D6 star 2, this is present in about 25% of Caucasians, about close to a third of Africans, but it's much less common in people from East Asia. But this is associated with an extensive metabolizer phenotype, star 2. Then they picked on 2D6 star 10. This is a very common allele in Asian populations, and it usually confers an intermediate metabolizer phenotype. So these folks can't metabolize certain substrates as well as others. And then on the end of there, 2D6 star 17, this is virtually only ever found in people of African ethnicity, and it's also another intermediate metabolizer phenotype associated gene variant. Okay, so they went and expressed each of these 3 variant forms of 2D6 in the bacteria, and compared the catalytic activity of those 3 variants to the normal, so-called normal P450 2D6 protein. And they use venlafaxine, as I said, as their substrate, and you can see what 2D6 is doing here, and it does this quite a lot. It's taking that methyl group off the methoxy substituent on the ring, and it's generating sort of a demethylated hydroxy metabolite. So if you look at the star 1 here, this is the functional wild type protein. You can see churning away. So remember we did these Michaelis-Menten substrate velocity curves way back in first semester in my lab on with rat P450. This is the same sort of thing they're doing here, but they're looking at venlafaxine as the substrate, and you can see when you look at the enzyme velocity here, picomoles per minute per picomole cytochrome P450, good activity in the wild type protein. But these different variant forms all had reduced activity, and star 17 here, the one that's common in Africans, you can see very poor catalytic activity indeed towards venlafaxine, and then star 10 and star 2 alleles were sort of intermediate. Then, as I mentioned, they also looked at the susceptibility of each of these four 2D6 forms with regards to the capacity to metabolize venlafaxine, the susceptibility to inhibition by quinidine. The quinidine's are malarial, and a very strong 2D6 inhibitor, contributes to lots of drug interactions. But the cool thing here is that here's the wild type protein, and sort of moderately susceptible to P450 inhibition by quinidine. But the interesting thing is that a couple of these variant forms, or even the B form here, the star 2 allele, is more susceptible to quinidine inhibition. And then, interestingly, the star 10 allele, quinidine was much less effective as an inhibitor. So that's just interesting, isn't it? That not only do these genotypic variant forms of P450 2D6 vary according to their capacity to metabolize a drug to substrate drugs, but they also vary according to the degree to which they are vulnerable to inhibition by P450 inhibitors. And this is why this stuff is clinically relevant. Nowadays, when a new drug is approved by the FDA, companies have to report not only pharmacogenomic information on how do the pharmacokinetics of the drug change in common P450 groups, variant groups, but also they want to know this sort of information. Are particular isoform-specific inhibitors more or less active in people with particular pharmacogene variants with the drug you're trying to get the FDA to approve. So it's making drug approval quite a bit more complicated in a way, because you've now got to not only look at the pharmacokinetics in common P450 pharmacogene groups, but you've also got to look at inhibitors of different P450, see if that varies. Now, okay, so that's all very interesting. So some of these variants are less able to metabolize venlafaxine, some of them are more or less susceptible to inhibition by quinidine. But the question that we're interested in as scientists and pharmacologists, of course, is why is all this happening? Why are some of these P450 variants less able to metabolize the substrate? And so they plotted the location of the variant amino acid in these various three common allelic variants with 2D6. And the interesting thing they found is that these variations are typically occurring in the substrate recognition sites. Now, if you remember my lectures way back in first semester, and I'm sure you don't, but remember that P450s typically have about 6 substrate recognition sites, and they're not as critical as, say some enzymes that you'd learn about in biochemistry, but essentially these amino acids just are important in binding to drug substrates and orienting the drug and stabilising it so it can get zapped by that all-important heme group at the bottom of the P450 carrying out the oxidation on the drug. Okay, so the point is that some of these common forms of P450 2D6 have variations in some of these substrate recognition sites that affect the ability of the protein to metabolise drugs. Okay, so if you look then at the alleles involved in these 3 variant forms, you can see that, like I said earlier, sometimes these variant P450s have more than one allele involved in variations in more than one allele within the particular protein. So what's going on here? We can see this with star 10 here. There's a P to S substitution at site 34 in the protein, a S to T at position 486, and these basically confer changes in the folding of the protein. We won't go through all of these, but all the others basically affect the substrate recognition site, all of them. Some of them occur in these critical helices or beta sheets that occur across multiple alleles. So sometimes some of these variant forms of the protein have a couple of these occurring within the same protein in this form. Okay, so what's going on in terms of P450 2D6 structure then? Well, most of these clinically significant polymorphic variants have more than one alteration with amino acids in critical sites. 6 star 10 here, you can see there's a change here, position 34, and then also 2D star 10 allele also has this S to T substitution at position 486. 17, star 17, that common one in Africans, you can see there's a position change up here, amino acid 107. There's also another change here at 296, and then there's a third change as well. Okay, so it's a point that some of these polymorphic forms of the P450 have several amino acid alterations at critical substrate recognition sites in the protein. And you're probably thinking, well, who cares? You know, does it really matter? Well, when they looked at the structure using X-ray crystallography of these proteins, actually they did it in silico, but they modelled the dimensions of the protein, and you can probably tell just by looking at it that compared to the star 1 common wild type form of the protein, which is fairly spacious protein, all these other variants, and particularly the star 10 and star 17 versions, give us smaller, more compact proteins. And that's going to be important because it means that it's probably going to be harder for substrates to get into the protein and get metabolised. That's basically the idea. And so they measured some of the physical dimensions of critical sites on the P450. And remember from my lectures in first semester, the active site volume of a protein is a P450 protein is very important. So they calculated the cubic angstroms of the active site within the P450 protein. You can see these variant forms considerably smaller active sites compared to the wild type star 1 allele. But then also the opening of that channel. Remember, the substrates have to get through the substrate channel to get into the all-important catalytic site. And you can see considerably smaller, narrower in the alleles, in the mutant alleles. So this African form here, down here, considerably narrower than in the wild type protein, just due to some of these amino acid substitutions occurring. And then the middle portion, sort of an indication of the spaciousness of the interior catalytic site, again, significantly more constrained in the couple of these variant forms compared to the wild type protein. So basically it just tells us that these polymorphic variant forms of 2D6 just produce a protein that's smaller and more compact, and that fewer drugs can get into the active site via the substrate channel. Okay, so Again, this has implications, like as I was saying in my lecture the other day. A lot of the early work and in the field of pharmacogenomics has been done in European populations or people of European ancestry in the United States and elsewhere. And the prevailing P452D6 alleles in those populations aren't always representative of people who come from different ethnic backgrounds. And if you look at the distribution of 2D6 alleles, there's very significant differences across ethnic populations. So this is why one of the reasons why 2D6 is so significant clinically. You can see East Asians over here. This star 10 allele is actually so common it's almost the wild type status in people of East Asian ancestry. Also significantly more common in people of South Asian ancestry, whereas in Europeans it's that little slice is just 1 or 2% of total population. So it's just the extreme form, but it's just making the point that we can't always assume that two people will have the same pharmacokinetic, or a given drug that's a substrate for 2D6 will have the same pharmacokinetic properties in people with different ethnic backgrounds. Okay, so that's just making the same point here that there are significant geographical variations across each of those four phenotypic groups of 2D6, the intermediate, null function, extensive metabolizers and poor metabolizers, and there are significant variations across different ethnographic sort of backgrounds. Okay, so this is one of the things that makes pharmacogenomics an important topic that we should be paying attention to, because it's a factor that contributes to variable drug responses in human beings. Okay, so, and that's the bottom line to take away there, that because of these 2D6 variant forms having different molecular features, different capacity to metabolize drugs, different capacity to be inhibited by different inhibitors, means that we can't assume that everyone's got the same metabolic capacity. We may need to make dose adjustments for certain drugs in people with PM or ultra-metabolizer genotypes, phenotypes. Okay, that's about it. I think I'm out of puff, but thanks very much for your presence, and I hope that got you interested in clinical relevance of pharmacogenes. In the next lecture we'll look at transporters as also vulnerable to this phenomenon. Thank you very much. Oh, sorry. Oh, sorry. Yeah. Oh, how'd you go? Yeah.
It had quite a high average. I think it was 21 out of 27 was the average, which is 78%. So when are we expecting the grades back for our presentation?
Next week.
Next week. Cool. Well, we go. Good.
Yeah. I think. No, I don't think it'll be very well.
Awesome. Great. Happy to do it.
see Maya? Did you see her group?
No.
They really took the corporate thing really well. They had colour-coded slides,
Yeah.
green and sort of yellow khaki gold, and they wore clothes to match. But they're really into the corporate
Yeah,
idea.
fair enough. Sure.
Some of the names I thought were outstanding. Better like this company I'm involved in, the Americans, you know, our name's okay, but we should have got students though.
Yeah, absolutely, yeah.
Some of them just fantastic.
Yeah. Alright, if it's not next week, the week after, we'll see you.
Yeah.